Hello everyone, welcome to my channel. Today in this video I am going to discuss about mitosis. So in the previous video I have already discussed about the interphase, that is called the preparatory phase or preparation phase. In that we have seen the G1 phase, S phase and G2 phase. In the G1 phase is called the growth phase, S is called the DNA synthesis and G2 is called growth and preparation for mitosis. So we have already discussed about that. This is the G1 phase, G1, S phase, G2 phase, and this is M phase or called the mitotic phase. From G1 to G2 is called, from here to till here, G1 to G2 is called the interphase. It is interphase and it is called the preparatory phase so that it helps in the cell division. So in this S phase is called the DNA replication phase. This phase is called the DNA replication phase, and G1 is called for the protein synthesis. This G1 is called protein synthesis and G2 is called growth and preparation for mitosis. This S phase is called the DNA replication phase. So this all I have discussed in the previous video. Now today I am going to start mitosis, this M phase. This mitosis I am going to start today. So the mitosis consists of two processes, that is karyokinesis and cytokinesis. Karyokinesis means division of nucleus. In this, nucleus divides. Cytokinesis means division of cytoplasm. Cytoplasm divides. So what are the phases of karyokinesis? There are four phases, that is Prophase, Metaphase, Anaphase and Telophase. So we will study all these phases one by one. The actual cell division will take place in the mitosis. The cell use the constituent and cell divide into two daughter cells. So what happens in karyokinesis? There is a first phase is called Prophase. Second one is called Metaphase, third one is Anaphase and fourth one is Telophase. So we'll see first Prophase. Prophase, which is the first stage of karyokinesis of mitosis. So this phase is called the first phase of mitosis. Follows S and G2 phase of the interphase. See, this prophase is divided into two phases, that is early prophase and late prophase. First is early prophase and the second one is late prophase. Now first I'll discuss early prophase. See, this mitosis is called equational division. Here I am writing this mitosis is called equational division. Equational division. Why it is called equational division? Because the chromosomes divide in both the daughter cells will be equal. Equal number of chromosomes will be present in both the daughter cells. So prophase is the first phase of mitosis, and in this there are two phases in this, that is early prophase and late prophase. So I am going to discuss early prophase. What happens? See, in the cell, what happens? A cell in the interphase, S phase, what happens? There will be a DNA replication will takes place inside the nucleus, and there will be a replication of DNA will takes place inside the nucleus in S phase. And after the replication of DNA, this cell will go for cell division in the M phase. Will go for cell division. So first step is what happens in the prophase, early prophase. This chromatin fiber start condensation. It start condensing in the early prophase. It start condensation, and the centrosome which undergoes for the duplication in the S phase of interphase now begins to move towards the opposite poles. This is centrosome gets duplicated and starts moving towards the pole. This is the aster, formation of aster, which will help in the formation of spindle fibers, and these spindle fibers get attached with the chromosomes during the cell division. So this is the early prophase, and in the late prophase what happens? In late prophase what happens? The nuclear membrane starts degenerating. This nuclear membrane will start degenerating, and the condensation of chromosomes will take place. The complete condensation of chromosomes will take place in this late prophase. This is centromere. This is centromere. So what happens in the late prophase? The nuclear membrane starts disintegrate, starts disappearing. Chromosome will be seen in the shapes. Condensation completes in this phase, and the cell organelles will also disappear in this phase. Whatever cell organelles will be present in this will get disappear. So what we see in this early prophase, the chromosomal material condenses to form compact mitotic chromosomes. The chromosomes are seen to be composed of two chromatids attached together at the centromere. See, this is a chromosome. These are sister chromatids. After the duplication or replication of the DNA, this is a single chromatids, and the duplicated chromatids will attach with this, and it will become a sister chromatids. Sister chromatids. So this and this will not be a sister chromatids. These are homologous chromosomes will be there, but these two will not be a sister chromatids. Non-sister chromatids. This is centromere through which this sister chromatids attached. So what happens in the early prophase? So we have seen the chromatin fiber condenses and the chromosome starts condensation. In early prophase we can see cell organelles disappears. And what happens in the late prophase? The nuclear membrane disintegrates, starts disappearing, and chromosomes will be seen in the shapes. Centriole moves. The centriole will, this is a centriole, and it will move towards the pole. Chromatin fiber condenses in early prophase. Second point, we can see cell organelles in early prophase. And what happens in the late prophase? The nuclear membrane disintegrate, starts disappearing. We can see chromosomes in shapes. Chromosome in shape. The chromosomal material condenses to form compact mitotic chromosomes. Chromosomes are seen to be composed of two chromatids. This is one and this is the second, attached with the centromere chromatids. Centrosome which had undergone duplication. This is centrosome, which get duplicated during interphase, begins to move towards to the pole, opposite pole, and it radiates microtubules. This radiates microtubules. This is a microtubules, or you can say aster, which helps in the formation of spindle fiber, and this spindle fiber get attached with the chromosome and they forms a mitotic apparatus. At the end of the prophase, when all the cell organelles will disappear in this phase. Cell organelles will disappear in this phase. Next, we will see metaphase. So what happens in the metaphase? All the chromosomes will be scattered in the cytoplasm. All the nuclear membrane will get disappear. Nuclear membrane will disappear. All the chromosome will be scattered in the cytoplasm, and they align together, align the chromosome in metaphasic plate. What is metaphasic? Metaphasic plate. Meta means second and phase means stage. This is the second stage of cell division. What happens after when the nuclear membrane will disappear, the chromosome will appear on the metaphasic plate. This is the metaphasic plate. Here is the centrosome. After the disintegration of nuclear envelope, this chromosome will align together in the metaphasic plate. This is the metaphasic plate, and see what happens in this. This is a chromosome and attached with the sister chromatids, and this is centromere. This is centromere by which these two sister chromatids are attached. This is centromere, and this is kinetochore. This kinetochore is disc shaped through which the spindle fiber attached. This spindle fiber get attached to the kinetochore. This is centrosome. This is centromere. This is kinetochore, and this is spindle fiber. And this is called metaphasic plate, on which the chromosome get aligned. So what we can see in the metaphase, the alignment of chromosome in the metaphasic plate, the spindle fiber get attached with the chromosome. So what happens in this metaphase? The disintegration of nuclear envelope and the chromosome will be spread into the cytoplasm, and they arranged in the metaphasic plate. Chromosomes contain two chromatids attached with the centromere. Spindle formation is complete, and this spindle formation will get complete and attached with the kinetochore. This kinetochore is a disc shape. All the chromosomes align themselves in the equator. This is the equator, or it is called metaphasic plate. Plane of alignment refers as metaphasic plane. Metaphasic plate. Centromere joins the two sister chromatids together. All the spindle fibers attached with kinetochore. First point is disintegration, nuclear envelope and chromosome spread into the cytoplasm. Second point is chromosome contains two sister chromatids attached through the centromere. Formation of spindle fiber complete. All the chromosomes align together in the equator. This is called equator, and this plane is called metaphasic plate. So all the chromosome will get aligned on the metaphasic plane, and the spindle fiber will get attached with this. And the next point is all the spindle fiber will get attached with the kinetochore. So next is Anaphase. Next is Anaphase. What happens in this phase? Chromatids moves towards the pole. This chromatids will start moving towards the pole. How it happens? By the shortening of the spindle fiber, it stretches towards the centromere. What happens in this? There will be a separation of chromosomes. There will be a separation of chromosomes will take place by the shortening of spindle fiber. By the shortening of the spindle fiber, this stretches, and both the chromosomes will get detached, and this chromatids will get detached, and now this detached chromatids will now called as chromosomes. Now this is a separate chromosome and this is a separate chromosomes, and it will start moving towards the pole. So actually this happens in the anaphase. Centromere, which holds the two chromatids, this is centromere which holds the two chromatids. These are the two chromatids attached with the centromere and get detached by the shortening of the spindle fiber, and they get split and separated daughter chromatids. Now these two are called daughter chromatids. These two will be called daughter chromatids, and it will start migrating towards the pole, opposite pole. Anaphase ends when the chromosomes reach to the pole. This happens, they are moving towards the pole by the shortening of the spindle fiber. First point is centromere, which hold the two chromatids together, splits and separate daughter chromatids. Next point is spindle fiber, shortening of the spindle fiber. Next point, chromosome migrate towards the pole. See this, chromosome starts moving towards the pole. Next point, this anaphase ends by the when the chromosome reach the poles. Ends when the chromosome reached the poles. This is called the poles. Next is telophase. So what happens in the telophase? Telophase is the end phase. The chromosome reaches towards the poles and the mitotic spindle fiber disappears. The spindle fiber will be disappear, and the decondensation of the chromosome will take place. Again the chromosome will decondense. There will be a reappearance of the nuclear membrane and the cell organelles like the mitochondria, Golgi apparatus, endoplasmic reticulum, all will appear, and the nuclear envelope will be again. What happens in this? Cell organelles will appear, reappearance of, you can see there will be a reappearance of nuclear envelope. You can say nuclear envelope or nuclear membrane. This telophase is same as the prophase. In this phase also there will be a early telophase and late telophase, and this is early telophase, and now next is late telophase. What happens in that? At the end of the telophase, the nuclear membrane will appear, and this chromosome will again decondense, and it will form into chromatin fiber, and the cell organelles will be in the visible, or it comes in the existence. At the end of telophase, nuclear membrane appear, decondensation of chromosomes, cell organelles comes into existence. So what we have seen in this telophase is called the end phase, and in this phase the chromosomes reach towards the poles, mitotic spindle fiber will disappear, decondensation of chromosomes, and the reappearance of the endoplasmic reticulum, Golgi apparatus, mitochondria, all the cell organelles, nuclear envelope or nuclear membrane will be visible, the reappearance of the nuclear envelope. And in this phase the chromosomes will get decondensed. So we have seen the prophase. What happens in the prophase? The nuclear membrane start disintegrate, and the chromosomes comes into shape. Therefore cell organelles will disappear in this phase, and in the metaphase it will come on the metaphasic plate and aligned and attached with the spindle fiber. In the anaphase there will be a separation of the chromosomes. Next we will see the cytokinesis. We have seen karyokinesis, now we will see cytokinesis. What is cytokinesis? There will be a division of cytoplasm. After the karyokinesis, this cytokinesis will take place. This is the end phase of the cell division and the division of the cytoplasm. In the animal cell, cytokinesis achieved by the formation of furrow. What is furrow? This is when the telophase, we have seen the shape of the cell like this. There will be a furrow, that is called the cleavage. This is called the cleavage furrow. And the cytoplasmic division will be takes place like this, and it is also called centripetal. Cytokinesis is also called centripetal because there will be a division will takes place by the cleavage furrow. This will takes place in animal cell, and it will get divided into two cells. Cytoplasmic division, cleavage. What happens in the animal cell, in the plant cell? In plant cell, formation of cell plate. In plant cell, this kind of cell division will not take, this kind of cytoplasmic division will not take place. In this there will be a formation of cell plate. Because why? Because in the plant cell, why it is not centripetal? In plant cell what happens? There is a cell wall, and we cannot divide the cell like animal cell in plant cell because of the presence of cell wall. and it is very stiff. That's why we cannot make a cleavage or furrow in the plant cell. So there will be a formation of cell plate in between the plant cell, and this cell plate is formed by the Golgi apparatus present in the plant cell, and this Golgi apparatus will help in the formation of cell plate, and this kind of cell division is called centrifugal division of plant cell cytokinesis. Hope it is clear to you all. Thank you.